Society of Gene and Cell Therapies briefing on cell and gene therapy approaches for HIV. My name is Margarita Valdez-Martinez. I am the Chief Advocacy Officer for ASGCT. We're very excited to share with you an outstanding panel of experts and patient advocates on this really important topic. So without further ado, I'm going to hand it over to ASGCT's president, Dr. Paula Cannon.
Thanks, Margarita. So I'll pull up some slides and let's get going. Okay. All right, welcome everybody to this research briefing on cell and gene therapy for HIV. And we're doing this a couple of days after World AIDS Day, which is December the 1st every year. I'm Paula Cannon. I'm from the University of Southern California. And as Margarita said, I'm also the current president of ASGCT. Just as a quick background, ASGCT is the primary professional membership organization for everybody who's involved in gene and cell therapy. It's everything from scientists and clinicians and patients, their advocates, and all the other tiers of professionals, including from government, who are working to increase the number of gene and cell therapies that we have in the clinic. So why are we talking about cell and gene therapy for HIV? I thought I'd start off with some background about that. So first of all, as I'm sure you're aware, HIV is still this massive global pandemic with almost 40 million people worldwide estimated to be living with HIV or AIDS. And even in the United States, the numbers are still kind of eye-popping. There is 1.2 million people estimated to be living with HIV and over 30,000 new infections every year. And one of the numbers that always strikes me as important is the fact that for every 100 people with HIV, only 87 of them know that they have HIV. So there's still a lot of room to be done in terms of getting people tested, knowing their status, and getting them into care. And the reasons for that are, the good news, if you like, is we have some very effective antiretroviral therapies, antiretroviral drugs. For example, to date, the FDA has approved almost 50 drugs from 10 different classes, and they're kind of shown on this very busy chart here, and most of them have unpronounceable names. But the good news is that nowadays, most people can take just one pill a day that combines 2 or 3 of these drugs into a very effective cocktail. And these drugs not only can treat HIV, but they can also prevent HIV if they're taken by uninfected people. And this is something that's called PrEP, again, 1 pill a day. And new innovations are constantly being rolled out, including long-lasting injectable formulations of the drugs, which are even easier. And so we are now at this amazing state that for people who can get these drugs and can take them regularly and tolerate them, their HIV can be kept at undetectable levels, and they basically cannot transmit the virus to anybody else. So this has been an amazing success from the pharmaceutical industry. But it's not all good news. And this little graph kind of shows one of the problems we face, that again, for every 100 people with HIV, 87% of people know that. But only 57% of people actually, get prescribed the drugs, stay in care, and the drugs work well in them so that their virus is stably suppressed. And these drugs do need to be taken every day for the rest of your life. And so because of these issues, we think there is a need for better treatment still, and even for a cure. Okay, so I talked about a cure. So I think it's worth just spending a couple of minutes on why we can't currently cure people who are living with HIV despite these amazing drugs. So the way I think about it is I think of HIV as being a triple threat virus. So first of all, it has this crazy high mutation rate. I mean, I think everybody knows about that viruses mutate from what we've seen with COVID and all the different COVID variants, and how we have to keep now changing the vaccine you get from COVID because the virus is mutating. But HIV makes COVID look like an absolute beginner. It has massively higher mutation rates than COVID. And these mutation rates happen in an individual person, so that even as somebody mounts an immune response against HIV and starts to make, for example, antibodies, the virus just mutates away from them. It's like a chess game. and the virus is always one step ahead. The second thing HIV does is that it infects and kills an important part of our immune system called the CD4 T cell, and that's shown over here. And CD4 T cells are like the conductor in the orchestra of the immune system. And so eventually over time, when they all get wiped out by HIV, this leads to a broad immune deficiency that then leads to these symptoms that we call AIDS. And then the final trick that HIV does is that it's a type of virus that when it gets into a CD4 T cell, it inserts itself into the DNA of that cell and becomes like a permanent passenger in that cell. And in some cases, the virus becomes latent. It basically goes to sleep. So our body doesn't see it, the drugs don't see it, and that would be fine, except that sometimes the virus can then wake up again. So if somebody stopped taking antiretroviral therapy, any virus that then reawakens will restart the infection. And that's shown in this little cartoon here, which is showing the levels of HIV in the blood of somebody. If they start on antiretroviral drugs, it plummets down to these almost undetectable levels, but stopping ART leads to this very rapid rebound. So these three things together combine to mean that we can't currently cure people of HIV. Okay, so I just lied, because there are actually, we think currently 7 people who've been cured of HIV. I call them the HIV unicorns, and I want to introduce them to you. So first of all, we had Timothy Ray Brown, who was also known as the Berlin patient. You may have heard of Timothy, and he was the first person to have been cured of HIV. And subsequently, here are some of the people who have kind of come out and speak to the media. Adam, the London patient, and Mark and Paul, the Dusseldorf patient and the City of Hope patient. And we're actually very lucky to have Paul, the City of Hope patient, on our call today. And he and I are going to be doing a little interview at the end of these talks. So what made these people unicorns? Well, first of all, the one thing they had in common is that as well as having HIV, they also got blood cancers. And as a result of their blood cancers, they needed to have what we call a stem cell transplant or a bone marrow transplant to help them fight their cancer. And the reason for that is that for these cancers and the chemotherapy that the guys were taking, that chemotherapy also kills your immune system stem cells. In other words, the stem cells that constantly make your immune cells. And so if those are wiped out by the chemotherapy, you lose your immune system and you would very rapidly die. And so we mitigate that by giving people a stem cell transplant from what we call a tissue matched donor. And that then creates a whole new immune system in their bodies that comes from the donor. So here's the trick. About 1% of people in the world are naturally immune to HIV, and that's because they have a harmless mutation that we call CCR5 delta 32. And CCR5 delta 32 removes a molecule called CCR5 from the surface of the CD4 T cells. And CCR5, along with CD4, creates the lock that HIV's key has to insert itself into in order to be able to infect the CD4 T cells. So for people with CCR5 delta 32 who don't have CCR5, HIV basically can't infect their cells. And so what was special about our HIV unicorns is that when they got their stem cell transplants, they didn't just get a stem cell transplant from a tissue matched donor, but they also got them from a tissue matched donor who happened to be one of these CCR5 delta 32 mutant people. And so as a result, the new immune systems that developed after their cancer chemotherapy and stem cell transplants, like their donors, were now resistant to HIV. So this is kind of crazy and extremely rare. Obviously, we wouldn't give a bone marrow transplant to somebody and chemotherapy to somebody just because they had HIV. This only happened as a side effect, if you like, of the cancer treatment. So I thought in the last couple of slides, I'll just introduce to you some of the ways that cell and gene therapies, that we're thinking about how cell and gene therapies could be used against HIV. So starting off with some really basic stuff, just, what is a cell and what is a gene? You know, we have billions and billions of cells in our body, and all our cells are a little bit different. But in the middle of the cells is where our chromosomes are, which are just long, long strings of DNA. And a gene is just a particular piece of that DNA that has characteristics that give you your eye color or your ability to smell asparagus and things like that. Okay, all the different traits that make us human. So when we think about gene therapy, a simple definition is that gene therapy can be used to repair a defective gene, modify a gene, or even give a whole new therapeutic gene into a cell. And one way we can do this is by directly injecting into the body our gene packaged into something that we call a vector, which delivers the gene into cells in the body. And you're going to be hearing after me a talk from Matt Gardner about how AAV vectors can be used to deliver anti-HIV antibodies as a potential gene therapy for HIV. And then in contrast, cell therapies, well, here we're not just putting a gene in, but we're putting whole cells into people. And they can include both unmodified cells that come from a donor, such as the donor stem cell transplants we were just talking about. Or we can take cells, certain cells out of a person's body, for example, their blood cells. We can modify them outside the body with our gene therapy vectors, and then we can return them into the body. And in that way, people can be given genetically modified cell therapies. And you'll hear about an example of this based on CAR T cells from our third speaker, Boro Japrolik. So finally, let's just a high-level view of some of the cell and gene therapies that are being developed for HIV. So first of all, we are using gene therapies to mimic these CCR5 delta 32 stem cell transplants by giving people a self-transplant. And so the idea of this is that rather than receiving cells from a donor, from a CCR5 delta 32 donor, which would be hard to find. Again, you have to have a tissue matched donor, which sometimes is very, very challenging. And then they also have to be the 1% of people who are also CCR5 delta 32. Instead, what we do is we take a patient's own stem cells out, we engineer them, in the lab to be CCR5 negative, and then we put them back in. So that's one way that a cell and gene therapy is being used. And then other ways are, we are thinking, well, we have an immune response against HIV, but it's not good enough to clear the virus. So can we improve our natural immune responses? And there's a couple of ways we think about doing this. Again, one of the ways is that we're using gene therapy vectors to deliver better antibodies against HIV, and Matt will be talking about that. Or we can engineer what we call CAR T-cells that are better able to kill HIV-infected cells in the body, and Boro is going to be talking to you about that. Okay, so that's the end of my sort of big picture introduction. Hopefully it's given you a good background, and I'm happy to introduce our next speaker, Matt Gardner. Okay, Matt.
Thank you, Paula. That was a great introduction to the topic. And I'm Matt Gardner. I'm an assistant professor at Emory University. I'm really excited to be here today to present to you a brief snapshot of one of our strategies that we're using gene therapy for an HIV therapy or cure research. On the next slide, basically, as Paula was hinting at, our work revolves around using this adeno-associated virus vector. Part of the reason I'm here today is because maybe about 20 years ago, I learned about this vector in high school. And I thought it was super neat that it's a virus that can be modified. Instead of infecting people and causing disease, it can be made into something that could be a new wave of medicine. And lo and behold, 2 decades later, I'm here now talking about this today. So why is AAV cool in my eyes? Well, one, it does not cause disease even naturally. It's just a small virus that encodes 2 viral genes, or what we call the virus parts, that are highlighted in the red and blue regions of the viral DNA genome that's packaged inside the shell of the virus. What we do with that genome is we gut it. We basically remove all the viral parts from that DNA sequence, and we place a therapeutic cargo in the middle of it that then ultimately gets packaged into the shell. And then we can harvest and purify the viral vector then, which can then be used to deliver our therapy into people. And so how this works is basically like there's a lot of different types of AAVs out there. They can enter all different types of cell subsets based on which AAV you're using. Well, all AAV does is it's really good at getting DNA into the nucleus of your cell. And what this does is it delivers the DNA contents that are packaged in the shell, delivers it to the nucleus, and it forms these circles, or called our AAV episomes, that can then, the cell can make the therapeutic cargo from. Importantly, this does not mean that it is modifying your DNA at all. It doesn't enter or insert itself into the chromosome like Paula said HIV does. It just sits there as a circular piece of DNA, and it can last for as long as the cell lasts in your body, making whatever therapy it is. So there's actually some evidence out there that this kind of therapy could last for years, if not decades long, which is a very key component for why we use AAV as our vector system. Next slide. So what is this therapeutic cargo that we encode within the AAV vector itself? Well, we are providing really good HIV antibodies. They're called broadly neutralizing antibodies. And in a typical infection of most pathogens like viruses and bacteria, humans will make antibodies that can help clear out an infection in about 7 to 28 days. As Paula said, HIV is a really good escape artist, and it keeps mutating very rapidly, and the antibody response cannot generate good enough antibodies that can suppress infection naturally. However, starting around 2009, a lot of groups were identifying these broadly neutralizing antibodies are really good, really potent antibodies against lots and lots of different types of HIV, different isolates of HIV. The problem was that these antibodies typically only developed years after infection, and that infection needed to persist. So basically, the people that were participants in these studies were not on ART during this. So it's not an ideal way of trying to generate these types of antibodies. One way that some groups are trying to do this is through vaccine design, similar to how we did for COVID. However, we still do not have a regimen in place that results from you going to the doctor and getting a shot that results in these really good broadly neutralizing antibodies. So this leads to our solution, and that solution is using AAV vectors that are then injected into your muscle cells, your muscle tissue, similar to a vaccine actually, and it turns your muscle cells into antibody-producing factories. So on the next slide is just a kind of a comic way, or a comic slide way of looking at this, where you take the AAV vectors and inject them into your muscle cells. Your muscle cells are those factories, and those factories are just bursting out with antibodies like your immune system would do naturally. And then over time, those antibodies are persisting in the blood. They can get to all certain tissue compartments where HIV can hide. And the thought is that with these antibodies there for years, if not decades, you can prevent new cells from being infected, but importantly, use those antibodies to engage your immune system to kill off infected cells as well. So we like to try to sell this as the possibility of a one-shot cure strategy, because based on the information we have, when this works, you go in, you get your injection, and you hopefully might never have to come back to the clinic for any more therapy after that. So on the next slide, how do we actually evaluate this as scientists? So we primarily use a monkey model. Monkeys are probably the gold standard animal model to evaluate HIV therapies that does not involve human clinical trials. Monkeys have very similar immune systems to us, and there are monkey forms of the virus called HIV or SIV that can infect monkeys naturally. And so what we do to study these AAV gene therapies in the monkey is we typically infect them with a HIV or SIV, which is then graphed accordingly over time in black to the amount of virus we can measure then in the blood. After a certain amount of time, the virus infection takes hold, And what we can do then, if you click next, with those monkeys is we can do two things. One, we can just let the infection persist, or we can use drugs to suppress the infection as highlighted in red. Basically, using antiretroviral drugs, like a lot of individuals are on and taking to suppress their infection, mimics a real world situation as well. And so after we're able to either let the infection persist or suppress the infection, we can then come in with our AAV vectors that make these antibodies, and we can test whether the viral infection either goes down, similar to antiretroviral therapy, or stays suppressed in the monkey without antiretroviral therapies present. And that is highlighted in blue. So success in this would be able to see similar results that antiretroviral therapy can do. However, we do not then use it in combination with antiretroviral therapy. And the monkeys allow us to study this for years. Like a lot of these therapy studies can take 2, 3, 4 years long. So it gives us confidence in the therapy. And there are actually even some examples now in publication from other groups that have shown that this strategy actually has legs and can work in the monkey model, suggesting that it could be an alternative antiretroviral therapy for humans, or possibly a curative solution, which would take more time for us to study. So I view this as a very simplistic model, and it seems like it should, hey, this is great, it should work. Well, unfortunately, there's a couple steps that we still need to solve in order to make AAV a viable solution for HIV therapy. And first and foremost is improving the consistency. I, along with a lot of other groups, have been testing this for a number of years now, and we do not have consistent results. There's always like a couple monkeys where you see good results, but the majority of monkeys, it does not work for a lot of reasons that we're trying to improve upon. But numbers 2 and 3 here, manufacturing and cost. How do we scale this to human therapy? Basically, we need to really work on the infrastructure to be able to generate batches and batches of AAV vectors that can deliver these antibodies for human use. And that is a costly endeavor. A lot of the FDA-approved AAV gene therapies that are already being used by people are very expensive, and it's just not something that would be easily to then have millions of people that are living with HIV have access to. So we really have to look in the big picture at solving some of these more difficult issues besides just showing that the solution can work in the lab. So I'm just going to stop right there. I do want to highlight a lot of other NIH-funded researchers that I have been working with for over a decade now at this point. But I also want to give a major shout out. As I said, a lot of this is dependent on the monkey research that we do. And I think all but one of the national primate research centers in the United States is now involved in this type of work. It is a very popular strategy. A lot of people are really interested in it, and we would not be able to do this without the help and support from our primate research centers and the staff that are there that do a great job handling the animals, taking care of the animals, and just doing just great work with the animals that are there. So I'll stop right there. I'll hand it off to our next speaker, Boro, who's going to talk to us about CAR T-cells.
Yeah, hello. My name is Boro Dropulic. I'm the executive director of Caring Cross. Caring Cross is a nonprofit, and its sole mission is to make these therapies affordable and accessible around the world. We work with stakeholders like hospitals and government-affiliated organizations to really reduce the cost of these therapies to make them really affordable and sustainable over the long term. And what I'm going to be talking about is how to engineer CAR T-cells that specifically kill HIV-infected cells. This is one of several programs that we have that we are developing. Next slide, please. This slide shows you the success of these cellular gene therapies. On the left, you have the CAR T-cell therapies. They're the approved products. They are targeting diseases like leukemia, lymphoma, multiple myeloma. And on the right there, you have stem cell gene therapies that are targeting various genetic diseases like sickle cell and beta thalassemia. Now, the big advantage of these therapies over the current drug therapies is that they are one-time therapies. And this will be much more economical over the long term than current drug therapies. And I believe that we're at the beginning of a new era of these transformative therapies. So that we're going to get better and better and have higher cure rates as we learn and improve these therapies, because we're taking cells from patients and modifying them and putting them back in. Next slide, please. And that's what this slide shows, that these are living medicines. These are one-time, long-lasting therapies from the patient's own cells. And on the right there, you'll see the general method that we use in order to deliver these therapies. We take the cells from the patient, then we add a gene to the cell, which is packed in a vector, and generally a lentiviral vector for these types of cells. And then these modified cells, whether they're T cells or stem cells, are then infused back to the patient. And this time period can be a couple of weeks, but we're rapidly moving. We're having advances that this can even be done in a day. That's how we are developing these technologies. And these are very specific, that they can address many chronic diseases. As you can see there, leukemia, lymphoma, multiple myeloma. This talk is focused on HIV, but it can also address sickle cell, beta thalassemia, various genetic diseases, and even fantastic data for the treatment of autoimmune diseases. So a wide platform for the treatment of many chronic diseases. Next slide, please. And this is the way that CAR T cells work. T cells are our white cells. They're our white cells in the blood. And so we take these white cells and isolate the T cell component of these white cells that's in green. And then when we modify them, then they express these CAR molecules on the surface. They're the squiggly lines that's highlighted there. And these CAR molecules, that is the payload gene that we delivered, can interact specifically with receptors on the surface of, say, a cancer cell or a HIV-infected cell, right? And then specifically kill that cell and then remove it from the body. So that's how CAR T cells work. So the same principles that we have had success in cancer, we are now applying for HIV, where the receptor is the molecule of HIV itself on the surface of the cell. And now this is data that we have generated that shows the promise of this type of therapy to develop it in terms of a cure for HIV. So on the left there, we have animal data where we have the plus C is the positive control animal. We won't go into the negative control animal. That's just an animal. That's the positive control has been infected with HIV, and you can see virus replication in the whole animal. Now we have two versions of the CAR that now those cells are being injected into the animal, and this is the ability to suppress the virus in the whole animal. A one-time therapy that has been delivered to the animal and completely suppresses the replication of the virus. So we're very excited with these results. We are now in phase 1/2 clinical trials, and we have already some very exciting data, but I can't disclose that in this forum because we're in the middle of a clinical trial. On the bottom, I want to highlight our partners like CIRM, UCSF, UC Davis, Case Western, and Einstein, who are partners in this endeavor with us. And the next slide. And this final slide addresses something that Matt was talking about, the cost of these therapies. These are one-time therapies, but at the moment they have a very high cost. You can see there that Kymriah, a product for the treatment of leukemia and lymphoma, $475,000. And then you can see that some of the products, the sickle cell product all the way to the right, $3.1 million. And the reason why Caring Cross exists is to address some of these cost issues, to make them affordable, that these one-time therapies can really be sustainable all around the world, particularly also in low and middle income countries. And we have a strategy to reduce that price tenfold from the current cost. And the way that we do that is by two axes of focus. One is lowering the material cost, and then two is local manufacturing at the hospital. So then we have a number of columns here where we're on the left there, you can see the green flasks there, low cost materials and equipment. Then the next one is short and simple manufacturing processes. When you have a short process, obviously the labor cost is much shorter, much lower than a long process. And then you have three areas there for point of care manufacturing. We believe that when you're taking cells out and putting them back in, the best place to manufacture these cells are at the hospital. So affordable facilities close to the hospital, economical logistics. And also when you make these products close to patients, you have better patient outcomes as well. And we are developing this model in concert with hospitals and government-affiliated organizations. There's no time in this kind of a forum to discuss about this, but if you're interested, my email credentials are right there, and I'd be happy to share that. And that's all I have to you today. Thank you very much, and I will pass you back to Paula. Paula, I think you're on mute.
Thank you. Sorry, I muted myself. I was being a good Zoom participant. In our remaining few minutes, Paul and I are going to be talking. Paul is the City of Hope patient you've already heard about, and we're going to talk about why these new therapies, and in particular an HIV cure, is important for people living with HIV. So I'll stop sharing my screen. Hello, Paul.
Hello, Paula. Thanks for having
you.
me here. You too.
Yeah, so it's always a pleasure to see me. So Paul, you're famous as the City of Hope patient now, and you're famous for being cured of HIV. But before you were cured of HIV, you lived with HIV for, I think, about 30 years. Can you tell us what that was like?
Yeah, well, I was there at the beginning of the HIV pandemic. It was a dark period. There was major stigma and lots of death. I lost count of how many friends and acquaintances I lost. The early medications were very difficult to take. They had a multitude of side effects. Basically, they kind of kept you sick, feeling sick all the time. Things did begin to get better in the mid-90s when protease inhibitors came along, and HIV medications have continued to improve since then.
Yeah, definitely. Yeah. So as I mentioned in my introduction, you developed a cancer, a type of blood cancer. I think yours was AML. Yeah, that was the cancer
Right.
you had.
Yes.
Yeah. And so when your doctors at City of Hope Hospital, this is where your name comes from City of Hope Hospital out here in Los Angeles. So when they told you had to have a stem cell transplant as part of your cancer treatment, but then they also told you about this kind of crazy idea of, shall we try and find you a donor who is a CCR5 delta 32 donor and potentially cure your HIV as well. What did you think of that when they first suggested it to you?
It was very exciting. There was no guarantee they would find a donor with the CCR5 variant, but they did. It was like looking for a needle in a haystack. They found a match, and it was I was kind of like holding my breath for a couple of years, not knowing whether it had worked or not.
Oh, so that's right, because I guess that they kept you stayed on the antiretroviral drugs even after you'd had the transplant, yeah?
Two
Yeah.
2 years afterward.
Yeah.
The plan was to stop a year afterwards, and the COVID pandemic came along, and I wanted to wait till I could be vaccinated for COVID, so we waited another year.
Yes, gosh, it must have been quite a decision point, because I guess that it's almost like kind of, you're kind of opening the box and you're asking, did this work or not? And yeah, so it must have been quite a difficult decision to know when to do that.
Right. Well, they had been testing me all along, and everything was coming up negative. So I felt good about stopping. I was ready after 2 years. But,
Yeah.
yeah, it was a little scary.
Yeah. So how has your life changed since you've been cured of HIV?
Well, I kind of feel like my legacy found me. I'm now on a journey of advocacy for HIV cure research. I've been going to HIV conferences and meeting with community groups around the globe, telling my story. It's very humbling experience, inspiring hope in people living with HIV and with the researchers.
Yeah, and we're so appreciative of you kind of coming out and being public about this, because you're no longer a private person, are you? You're kind of recognized on the street. So
Yeah.
um so so maybe I can ask you a question, Paul, that's a bigger question, and it's to speak on behalf of people living with HIV, and of course the vast majority who are not currently cured of HIV. I mean, I in my introduction, I talked about the fact we have these great drugs, you know, and so people sometimes say to me, what's the issue? You know, take a drug a day, you know. Why do people living with HIV want a cure?
Well, there are many reasons for a cure. It will free people from having to take medications for the HIV for the rest of their lives. It'll free people from living with the fear of transmitting HIV and free them from the stigma attached to HIV. It's also going to encourage people to be tested and treated. And a cure would essentially eliminate the uh, global HIV pandemic. It also drug resistance and paved the way for potential treatments for other conditions like cancer and Alzheimer's. You know, I didn't set out to be cured of HIV. My stem cell transplant was to cure my acute myeloid leukemia, and it cured my HIV as well. So I'm very fortunate. It was just an incredible side effect.
Yeah. I showed a photograph of you with Mark Franke, who's called the Dusseldorf patient. He's another person formerly living with HIV who's been cured, and you and Mark and also Adam, you're very gracious with your time and coming to meetings. You inspire scientists like me, you inspire clinicians, and I think you really inspire the rest of the community of people living with HIV that this is possible. And maybe it's, you know, I call you guys unicorns. You know, as we talked about, a stem cell transplant isn't at all for anybody who doesn't need it because of an underlying serious cancer. And it was certainly very grueling to go through. But I think, you know, we point to you guys and we say, look, you know, this might be crazy difficult, but it's happened. And now we want to start using these new medicines like cell and gene therapy and see if we can come up with something that can also achieve a cure, but, you know, far less grueling procedures to go through. Yeah. All right, Laura.
I was there at the beginning of the pandemic. I hope to be there at the end of it.
Oh, gonna make me cry. All right. Thank you so much, Paul. So Margarita, shall I hand it back to you, and you can see if there's any questions that people have put in the chat for us?
Yes, absolutely. Caitlin, can you add all of our speakers? Perfect, thank you. And for our congressional staff, please know that you are welcome to submit questions through the chat. You can submit them anonymously, so feel welcome to ask anything that you want, and you can also send questions or follow up. If you want follow-up information, we can send that via email as well. Paul, I just want to thank you first and foremost for being a part of today's briefing, and for, as Paula says, so generously lending your time. I think you inspire not only the amazing researchers that we have on this briefing, but also the layperson that's just excited to see these advances in medicine. I can imagine that it was a very pleasant surprise when you saw a cure. Can you tell us a little bit about what inspired you to actually go public? Because while it's a great benefit, there's also probably a lot of questions and a lot of burden. Maybe you had a private life before that you said, as you talked about the stigma. So now you're being very forward facing and sharing this wonderful benefit. What led you to that decision?
Well, I remember well when it came out about Timothy Ray Brown, and you know, it gave me hope that someday there would be a cure. So when this happened to me 30 years after living with HIV, I decided that it was an important story to put out and tell because it does inspire people. It inspires me as well. You know, you know, it's I'm really happy that I made the decision to go public.
Thank you, Paul. Continuing with the patient experience in that thread, Matt and Burrow, you talked about different approaches to therapies, in vivo and ex vivo. Can you talk a little bit about that and specifically what a patient could expect to experience with an in vivo or ex vivo therapy?
I'll start with the in vivo part. Basically, this is something where the therapy product is made, delivered to the clinic, and it's there waiting to be used. So as I think I mentioned during my talk, it would be very similar to a vaccination. It might be a few more different locations where you would receive an injection than just a single shot on the day that you go to the clinic. But basically, you would go in there, you would just receive a few injections into your muscle tissue and then leave. But maybe you would take a little bit of Tylenol to alleviate any soreness. But overall, I would then anticipate that probably over time, that individual would come back just to make sure, like, one, the antibodies are being expressed for at a decent level. But overall, like, as far as therapy would go, you would hope that after that one day in the clinic, that's the only therapy you would need for decades, if not your lifetime.
Yeah, and for the ex vivo, these are now standardized procedures where the participant or patient would come in. They would undergo what is called a leukapheresis, where you're taking out the white cells or whole blood. And then those cells are then processed. The patient would then go on a drug regimen in order to precondition. So that basically it creates an environment for the cells to engraft very efficiently into the body. And then the patient would come back a week later to receive his or her own cells, and then ready to and then simply monitored after that point.
Great, thank you. So I'll throw this out to the full group. Obviously, we've talked about a variety of different approaches. Are there any approaches, therapies that we have not talked about, even if they're just in the very beginning stages? Anything that you would want to share with the audience about other options out there in the pipeline?
Perhaps I can take that, because really there is just such extraordinary innovation, I think, in the cell and gene therapy world. And we're seeing innovation for cancer, for genetic diseases, as Boro talked about. And so there's some other applications that are being tailor-made for HIV. One example we can talk about is you may have heard of CRISPR, which is CRISPR is a type of gene therapy that has a high degree of precision. It allows you to go in and kind of actually find a specific gene and manipulate it. So, for example, I talked about how gene therapies could be used to find and kind of knock out the CCR5 gene. In addition, you can think about using CRISPR and similar gene therapies to seek out and destroy the HIV virus. Remember, I talked about how HIV is this weird virus that actually inserts itself into our own DNA. So in effect, it becomes like a genetic parasite in our body. And so companies are developing CRISPR-based gene therapies that could seek out and destroy HIV as it's sitting in our cells, especially these latent sleeping forms of HIV that are very, very hard to get rid of. So that's, kind of sounds like science fiction, really. But again, I think we'll get better and better at using these. There's Boro and Matt, do you want to? There's also other immune cells.
I think you're being a little modest too, Paula, because Paula's involved in as well as a number of labs these days in making what are called CAR B cells. So similar to CAR T cells, they're modifying natural B cells, which are the professional antibody making cells, to make HIV antibodies. So unlike what I do with AAV vectors to turn muscle cells into antibody factories, she's actually doing work to turn the natural producing antibody cells to make the HIV antibodies too. I think I'm being fair when I say it's also early stages, but there could be long-term products available through this kind of work that's not just for HIV, but for other diseases as well.
Fantastic. So, going back to the patient experience and kind of the patient perspective, what has the HIV community's response been to the potential for gene therapy or these other therapies? Are there frequently asked questions or concerns, or is there general excitement? What are things that you would like the audience to know that maybe their constituents may have questions about?
I can start because I had my eyes open to this about 8 years ago. The NIH funds these collaboratory grant mechanisms called the Martin Delaney Collaboratories. And part of those collaboratories, each large group that receives NIH funding for this work, they have to have a community of people living with HIV be part of that. And so my work in some of these collaboratories, I've been able to talk to a number of people living with HIV, and specifically, I get to ask them those types of questions. And for the most part, yes, there's a lot of very wariness around gene therapy, just the terminology in general. But there are a number of individuals, very selfless, courageous people that might not understand the science that well, but are getting heavily involved, trying to learn, trying to advocate for it too, that would like to see these different therapies come to fruition and be part of some of the clinical trials come into it. So while they have concerns about them as well, they do voice those concerns to us, and it gives us as researchers a different perspective because we're heavily involved in the science, but we also get to see if we're doing the science for this group of people, how can we do better in the lab to make it more available to them and easier for them to understand and less of a risk for them should they want to be involved in it. So I think like clearly not that maybe gene therapies aren't for everybody, cell and gene therapies for everybody, but I think there's a number of people within the HIV community that are looking for a real cure that traditional antiretroviral therapies do not give them.
Yeah, I would agree with that. There is a subset, if you like, of HIV-infected individual that are very motivated about seeing a potential cure.
And I think, and you know, Paul, you can probably speak to this as well, that people living with HIV they were kind of like the OG patient advocates, weren't they? You know, they sort of we saw through ACT UP and the refusal to be sidelined and ignored, the pressure that people with HIV put on the government to treat the disease seriously, to develop drugs, the selfless enrollment in clinical trials, and some of the early drugs were very toxic. And I often find, Paul, when I speak to people, especially from your generation who were around in the time before we even had the drugs we have today, this kind of selfless wish to give back still also, you know, be a little bit disruptive, be advocates still, and as you yourself said, and it almost made me cry when you said you want to be around for the end of this pandemic as well.
Yeah, it's been a hard fought pandemic, you know, but I definitely want to be around to see the end, you know.
Thank you, Paul. You're definitely inspiring all of us. You know, I also want to share with the audience that ASGCT partners with patient advocates and organizations to develop really robust resources that may be of help or assistance to your constituency. So I'll ask one of my colleagues to please drop the link to our considerations for clinical trials and some of our 101 materials, which we'll also distribute via email too. So this may be something that you'd like to share with your legislators' constituency. I also want to address one of the questions that we received in the chat, and I will open this up to the full group. Could you speak to the efficacy of current work in colorectal cancer and its potential implication for HIV prevention? I know some impressive work is being done at the vaccine branch at the NCI and buttressing the Liam of some types of cytokines. I wonder what kind of research would mean for HIV prevention and treatment.
I'm not sure I can speak about that specifically, but what I can say is that there's so much, you know, cancer is incredibly complicated, and in a way that's good news for us, because that means that as people are developing cell and gene therapies for cancers, they are also approaching it from multiple different angles. And in particular, there's interest in, just as we talk about this with HIV, can we kind of improve the immune system so that our immune systems can go out and neutralize HIV or kill HIV-infected cells? A lot of the basic concepts in cancer cell and gene therapy are also doing that. You're basically saying, if you've got cancer, part of the problem is that your immune system hasn't seen it and isn't kind of going and clearing it out. So can we make better cancer gene and cell therapies? Borrow talked about the CAR T-cells, which came from the cancer world, but are now being applied for HIV. And there's other things that are being done to kind of further ramp up the immune system, to recognize things that are different and unique about cancer cells, and just take the many different branches of the immune system and sort of focus them on a target. So while I don't know, and maybe Borrow or Matt, you do know in particular what's happening with colorectal cancer, again, I would just sort of say that a lot that work on HIV and cancer and autoimmune diseases and the genetic diseases is all going in parallel. And although today we're focusing on cell and gene therapy for HIV, really so much of what we're talking about is going to be applied very broadly.
Yeah, there's a lot of work in the CAR T-cell space, particular for colorectal cancer, but in cancer in general, whether it's colorectal or leukemia and lymphoma or other cancers, we are learning a lot on cells, how to make them more tailored, how to make them highly more potent. And all those learnings are collective. I mean, whether we're working in HIV or the cancer space, these all learnings synergize and work to create newer and better generation products.
Yeah, I mean, go ahead, Matt.
I was just going to say, I can't speak specifically on colorectal cancer, but being at Emory, there's a lot of work being done on understanding what's called the tumor microenvironment. So basically, what all is happening within tumors of a lot of different types of cancers. And the basic research that's going on there has informed us on so much of why does your immune system not be able to identify these as bad cells and clear them. And that, touching on Paula's point, is that we take that kind of information from those types of studies and we can directly apply it to how we understand why HIV persists. Because trying to clear out HIV-infected cells is like a needle in a haystack that has similar features to why can't the immune system clear out cancer. So there is, for a lack of a better term, crosstalk between the fields that we can definitely utilize some of the information just from the immune system in general for both types of studies, be it cancer or for HIV.
Yeah, all the different pieces. Again, sometimes we go in and we think, the immune system has these different, it has antibodies, it has the T cells that kill cells, but there's also other parts of the immune system whose job is to kind of like flag cancers or virus infected cells, or to present little pieces of that cancer or that virus to the rest of the immune system and kind of say, hey, come over here, there's a cancer you should be attacking, or there's HIV infected cells you should be attacking. So as cell and gene therapists look at this, the orchestra of the immune system, we see lots of opportunities for interventions, places where we could go in and we could say, let's make that a bit better. Let's make these CAR T cells a bit better. Matt talked about the work I'm doing making B cells to make antibodies. There is also people working on another type of immune cell called the natural killer cell. Such a cool name, NK cells. And they're taking NK cells and they're saying, go and find that cancer or go and find that HIV infected cell. So lots of, there's room for your imagination, if you like, as a scientist. And I think we're really at the start of this sort of golden age of being able to manipulate cells, especially of the immune system, to treat many diseases.
What I really like about the cell and gene therapy space and engineering cells is just that. It's very different than drug discovery. When you do drug discovery, you're panning and you're looking for a drug, and that few drugs could fail because of toxicity or because of pharmacodynamics. We have the whole nature as a toolbox. to continually engineer, right, and improve these therapies. And I've absolutely no doubt that in the years to come, we're going to learn so much that we are going to truly have therapies that are curative for whole classes of diseases, because we've got the toolbox of nature to help us to do that.
And I think the nice thing with cell and gene therapies in general is like maybe your body naturally can't fight these diseases like latent HIV and curing or even cancer, but we have the ability to basically say, look, these cells should be doing this. They're not doing it. Let's make them do it and actually be more therapeutic rather than just hoping that your body can naturally figure it out.
It's really like a spa treatment for your cells, isn't it? Let's go and rejuvenate them a bit, make them do this.
I have another question from the audience. A couple of questions from the audience, actually. I'll start with this one, and this is open to the full group. We are anticipating PEPFAR reauthorization in 2025. In my view, this is one of the most successful foreign policy tools that has been deployed by the US in the 21st century so far, and it has saved countless lives, including many of those living with HIV in the global South. Could you talk about how this program has helped in your work, if at all?
I think, Boro, do you want to talk about, because this really speaks to the fact that, I view this as one of the most, not just, how amazing it's one of the most selfless things this country has ever done, and bravest things in terms of deciding that they were going to make antiretroviral drugs available globally. Huge undertaking, 40 million people with HIV worldwide. But oh my gosh, it's bad as the pandemic of HIV is today, it would be so much worse if the United States hadn't decided to do that. So I think that ethos, that idea that we can be pioneers, and including with HIV and AIDS, and that we should be brave, but also that as we're developing these new therapies, it's really important that we think about accessibility and affordability. So Boro, I know Caring Cross does a lot of work in this space.
Yeah, that certainly has been highly impactful program, and it has saved countless of lives. It's one of the best things I think that we have done as a foreign policy. All I would just say is that we're at the early stages of gene therapy, but as these engineered cells or direct products become shown to be safe and efficacious, we certainly can then use those mechanisms to get these therapies out into those countries. And we're very much focused, Caring Cross is, as an organization, for how do we work in low middle income countries to get these curative therapies out there. And it's not just simply delivering a drug, it is more setting up infrastructure. When you're doing personalized therapies, you have to have, local facilities, you have to do technology transfer. We do a lot of that in terms of teaching other countries to be able to manufacture these therapies so they can manufacture at a much lower cost within the country. So for example, we have a partnership with Fiocruz, which is the large biologics manufacturing organization in Brazil that we're funded by the Brazilian Ministry of Health. And so it's those kind of initiatives, creating infrastructure and then being able to then provide support to produce these products locally, that I think will hopefully, layer on top of the fantastic work that PEPFAR has already accomplished.
Fantastic. I know we're getting close to the hour. We have, so this will probably be rapid fire responses. But I do want to address a couple of more questions that came in and open it up to the group. Let's see. In the presentation, Dr. Cannon, you mentioned many HIV patients do not know that they have it. And is that because there is a lack to access for screening for HIV?
I'm sure that a component of it is just ignorance that they could have been put at risk and are now infected. Maybe some hesitation, as well as lack of access. There, in most major cities, Paul can help me with this. It's relatively straightforward to find an organization that will provide free testing. But you know what human beings are like. Just because you can get tested, it doesn't always mean that people will. And so some of the work we're doing, not just to make better drugs, but also to potentially provide cures, I think will go some way to encouraging people to get tested. It's if you know there's a treatment and there will, you know, it's that encourages you. Yeah, Paul?
Yes, I totally agree. I think it will encourage people. Absolutely.
So we're right at the hour, but I just want to close this out and thank every single one of you for taking your time to be here. And then if we can just quickly go around the Zoom, and we'll end with you, Paul. If you can share one thing that you would like this audience to take away from this briefing, what would that be? And we'll start with you, Dr. Cannon.
Oh, gosh. Cell and gene therapies are amazing, innovative medicines, but they also make sense. And they're here, and we're going to see explosive growth in them, and not just HIV and not just cancer, but autoimmune diseases, Alzheimer's, everything, all these really tractable and chronic diseases that we don't have good solutions for. I am just so optimistic that cell and gene therapy is going to deliver.
Matt?
Just want to probably add on to that, these therapies are very exciting with the breadth of diseases that they could cover, that I think it's very important that we as a society stay invested in these kinds of research. It is expensive, but I think with time and interest, we can start to really hammer down the price and start really opening up the accessibility to some of these, what were used to be thought were too expensive for most people. So I think the future is really bright for this type of medicine.
Yeah, and again, I'll just add to both of those comments, is that very exciting future for this field. I think we will get to cures for classes of diseases, and we're already making inroads to decrease the cost. Everything that is new starts at a high cost, right? It's part of innovation, but we are learning how to reduce those costs, and that way we can make these living medicines that are one time really affordable over the long term, for the benefit of society.
And Paul?
Yeah, well, I think the gene therapy is very exciting and very important. I think it will probably lead to a cure for many diseases, like everybody's been saying. So I want it all to continue.
Thank you, Paul. Thank you, everyone. And thank you for the audience for attending and for your thoughtful questions. We will send a link with the recording and resources. Thank you again for your time and efforts. Bye, everyone.
Thank you.
Bye.